behind me is I actually defended my dissertation on. So I'm going to talk to you a little bit about an Archaic period archaeological site, and then how oysters that were excavated from that site reveal sea level rise thousands of years ago. See if this works. No. Okay. Must be a lag. All right. So the archaeological site, which was called Minnesota Key offshore, is located in the Gulf on the southwest coast of Florida off Sarasota County. Currently, it's about 1,000 feet off the beach in about 22 feet of water, and it was discovered in 2016 by a scuba diver who was looking for shark's teeth. Inadvertently, he found a human jawbone while he was on his dive. He reported it to my office. It's under our purview. And we noticed that the jaw was very robust and the teeth were very worn down. So that suggested to us that it was very, very old. We weren't sure if it was something that originated on the beach, which you can see in that map there where it says Minnesota Key, and then washed in to where he found the human jawbone, or if it originated from that location, which would be crazy because how could it survive in the ocean? So we went down there to investigate, and we found what looked like peat, which is something that is normally in fresh water on the bottom of ponds and lakes. So it's like highly organic, it's dense, that kind of like mucky stuff when you're running around in a lake. But that was actually coming out of marine sand, and inside of that peat was embedded human remains and worked wooden stakes. So this was like very surprising to us. Okay. So this, we automatically had an indication that this could be an example of a mortuary pond, also known as a charnel pond or a burial pond. And this is a cultural practice that is unique to Florida. Florida has specific environments that allow preservation of these types of sites. So it's unique to Florida. You can see the current examples of mortuary ponds on the screen. Wendover is the best known one. That one was actually studied archaeologically. The rest of them, Ryder Pond, Bay West, those were all kind of rescue archaeology efforts. So bulldozing was happening and people were trying to study what they could before it disappeared. But Wendover was actually archaeologically excavated. So most of what we know about mortuary ponds comes from one mortuary pond. They are characteristic, or the characteristics of these ponds are people were placing their deceased individuals into shallow freshwater environments. They would excavate little pits into the peat and then put branches that they had fire hardened on one end and sharpened and bend them over their loved one to either keep them in place or maybe mark the burials on the surface. We're not sure, that's conjecture. But either way, these mortuary ponds, there are human remains embedded in a watery environment with worked wooden stakes. And we had the same thing at Minnesota Key offshore. They typically date to the early and middle Archaic period. So the range of dates is like 10,000 years to like 5,800 years. Well, for the pyramids, which is kind of cool to think about, but most of them cluster around a 7,000-year-old date. So most of these mortuary ponds, they cluster around 7,000 years ago. To continue investigating the site, we had a bunch of questions. We conducted remote sensing survey, basically to build a picture of what it looks like. So the top one, that top photo, that's a sector scanning sonar, which basically gave us a view top down. So you can kind of see the peat coming out of the seafloor there. And there's also some scuba diver bubbles to the far right. But so we also did subbottom profiling that gave us a cake slice of what the sediments look like under the surface. So that's what that bottom picture is. You can see the pond basin there, which turned out to be a lot larger than we thought it was originally going to be. We also collected sediment cores to analyze the before the pond, during the pond, and then after the pond sediment deposition. And we also excavated 4 units. We did not target any areas that had visible human remains. But when we ended up excavating down, we did come across human remains. That's how dense this site was. Ultimately, we did confirm that, yes, it is another example of a mortuary pond. we had worked wooden stakes, and they dated to about 7,400 to 7,080 years old. So pretty remarkable. And I have some of those artists' rendition of what it may have looked like, what these burials may have looked like. But what we realized we ran into were most of the tops of the burials were just eroded away, because I mean it's in the ocean. You can't fight Mother Nature. So we really just had the bases of these burials. And the longest example of a stake that we had was like maybe that long. When from Windover, I mean they were meters long. So we didn't have that in our case. All right. So again, we confirmed that the site is another example of a mortuary pond, and it was used as a place of interment during the Middle Archaic period. So when the site was used thousands of years ago, sea levels were much lower. And I have a couple examples of ancient shorelines up here. So the biggest portion of Florida that you see up there on the screen, sorry, I don't have a pointer. That's about 20,000 years ago at the last glacial maximum. So that's when it was really cold. All the water was locked up in the northern ice caps, revealing twice as much of the Florida peninsula than we have today. So when this site was used as an actual freshwater mortuary pond, where it's located now, you can see the shoreline would have been like 9,000 years ago, actually. Up there, if you were standing where those people were interred, you wouldn't have seen the beach at all. So 7,000 years ago, you wouldn't have seen the coastline at all. So this was well inland when it was used. And now, due to sea level rise thousands of years ago, it's in a completely offshore context. And it's shocking that sensitive material, like you think bone and wood is very sensitive, could survive sea level rise, which we usually tend to think of as a catastrophic process. It's very dynamic. The coast is changing. Large waves are like coming closer. So the fact that this survived sea level rise was shocking to us, and we just had a lot of questions. And this is where my oysters come in. So I was in charge of doing all the artifact photos and everything like that. So I started to notice that a lot of these oysters had either had been attached to human remains or had impressions left on them that suggested they had been attached to human remains or even wooden stakes. So I'm not going to show any depictions of human remains, and none of this study did not involve any archaeological material or human remains. That has all been repatriated and reburied in a safe onshore location. So really what you see, the gray part, those are impressions of where material once was. So what does this mean? Well, the oysters must have come after the burials, obviously, but they were not recent growths either, because these were excavated from beneath the surface. You know, it's not like these grew in like 2014. Also, oysters need access to the water column in order to breathe and filter feed. If they become buried, they will die due to smothering. So once a young oyster settles, it's like an oyster larvae becomes a spat. Once it settles onto a surface and it cements to it and it never moves again. So it's not like these things are moving around. And they're most commonly found in brackish, estuarine, or near coastal waters with some sort of salinity. So not a freshwater pond in an inland setting. The oysters at the archaeological site offered me, in my studies, an opportunity to understand the slow swallowing of this once inland freshwater pond by the sea, and hopefully to gain insight into how a delicate archaeological site could survive for so long. Because everyone said there's no way that site could exist there. There's just no way, but here it is. There were also many instances of barnacles attached to wooden stakes and skeletal material. Other oyster shells had attachment scars or little impressions that looked like they could fit around wooden stakes or maybe larger long bones. In the interior portion of the site, some of these oysters were buried under at least a foot of sediment. So again, not a recent phenomenon. So we can glean from these oysters that thousands-year-old skeletal material was exposed just long enough for an oyster to settle upon it and then grow, who knows how many years. And then eventually the site was capped by sand. So the water had to have been saline enough to support the oysters. And then based on these oysters alone, maybe we can do some investigations. There's a unique opportunity to study past sea level rise thousands of years ago, and to do that, I used stable isotope analysis. I have zero background in stable isotope analysis, but this was the way I had to go, so I had to learn. So what is it? What does it even mean? What are stable isotopes? How do they work? How do archaeologists use them to infer information about human behavior? Isotope is a fancy word for elements that have almost the same chemical properties, but different numbers of neutrons. So in other words, it's the same element with a different atomic mass. You might be familiar with this, especially if you have a background in archaeology like me, with radiocarbon dating. That's basically using C14, the decay of C14, to determine the age of the sample, and that's an isotope of carbon. Isotopes are everywhere around us, and they're in the atmosphere, they're in the ocean, they're in plants, animals, they're in you. Some other examples that you might have heard of are like nitrogen, strontium, oxygen, which is what I'm going to talk about today. Isotopic signatures vary based on geographic location and conditions. So cold, fresh water up in those northern pole areas are going to have a different isotopic signature to warm, tropical, brackish water, near the equator. They also vary based on organism. If we look at plants and animals, plants are going to have an isotopic signature that's vastly different from an herbivore or an animal that eats a lot of meat. This is because stable isotopes are incorporated into body tissues through food and water, and that also includes humans. So, for example, there's projects where we look at human teeth to see where they're getting their drinking water from. So that's just an example of like the isotopic signatures that can be found in your body. And you can also tell if someone's diet is based heavily on corn or wheat or meat, which is pretty cool. Since oysters form in water, we're going to talk about water today. We all know the life cycle of water: precipitation, condensation, runoff, evaporation. And I already told you that isotopes are present in water. So those isotopic signatures depend on the water cycle and various physical and chemical reactions, which is known as fractionation, or the enrichment of one isotope over another. So in other words, it's the differential incorporation of heavy and light isotopes. In terms of oxygen isotopes, which is one of the ones that I used in my study, evaporation can take the lighter isotope first, which is O16, and leaves the heavier isotope behind, that's O18. So scientists can look at the ratio of O18 versus O16 to examine evaporation, precipitation, and even temperature changes in the water. It's a lot. Had to not memorize that part. So basically, to boil it all down, isotopes are everywhere and have specific signatures, and organisms take up isotopes. So to relate to the oysters, if there are specific isotopic signatures in bodies of water, then it would stand to reason that the oysters that are incorporating those isotopes into it, like their shell structures, are going to reflect whatever's in that body of water. And that's really, really convenient if you have oysters that were attached to skeletal material and wooden stakes, and you want to know more about the water body that it was growing in. And oysters form in isotopic equilibrium with the surrounding water. So it's like a 1-to-1. It's great. Makes my job easier. So where is this isotopic information stored? It's actually in the hinge of the oyster. So, you've got the top shell, the bottom shell that has all that delicious meat in it. You take the top shell off, there's a hinge, and that's where the isotopic signatures are. So basically, I had to bisect the hinge of these oysters, and this is what it looks like when you cut it open. So the top picture is the oyster that's been opened up, the hinge that's been opened up, and you can kind of see that there are little stripes in there, or growth bands, kind of like growth rings on a tree. So just like a tree will form a growth ring, I think it's like cold season and then warm season, oysters will do the same thing. And they're incorporating the isotopic data into those growth rings. So just as archaeologists can move back in time as we dig deeper and deeper in the sediments and peel back the layers, I can also go backwards in time if I'm moving through oyster growth bands. So the most recent growth, there's an arrow up there that kind of shows you the direction. So to move backwards in time, you go towards the very tip of the hinge, essentially. All right. So what environmental changes am I looking for when I'm looking at the hinges of these oysters? I'm basically looking at oxygen and carbon isotopes. So the ratio of O18 to O16, which is also known as delta 18O. I'm just going to say oxygen, but really it's delta 18O is that ratio. And then the ratio for carbon is C13 to C12, also known as delta 13C. I'm just going to say carbon just to make it easier. And with this, higher water temperatures, increases in freshwater, so like tons of precipitation, will cause more negative oxygen values in the oyster, so delta 18O values. And the opposite is true for if you have colder temperatures and more evaporation and more saline water, you're going to have more positive oxygen values in that oyster. And then for the carbon, more decomposition of organic matter, a lot of dying leaves and that kind of thing, and a decrease in salinity will cause negative carbon values, just kind of generally. Carbon's a little harder to interpret, but we're going to mostly focus on oxygen values today. So as oysters deposit new layers of shell, they're recording temperature and salinity changes in the surrounding water. So how did I get at this? To extract the data, I went to a lab in Alabama and I took my little bisected hinges, and because they're like teeny tiny, I think that one up there is like maybe a centimeter. Say, yeah, that's 1 centimeter. So they're really tiny. I had to load them into a machine with a microscope, and then I had to draw pathways where I wanted to basically scrape powdered calcite out of each growth band. So the more powdered calcite I collected, the more area I sampled, the bigger picture I would get, the finer detail I would get, because if an oyster is laying down a gray band and a white band every year, then I can see subtle changes in salinity and temperature as the oyster is growing. So that's why I wanted to use a microscope, and I had to use a dental drill to drill pathways, scrape powder into a glass tube, and then put it in a tray and do it 557 times. And all of that was run through a mass spectrometer in Alabama. And then, yeah, I got the data. But let's see, same like so I did this for 8 different oysters from archaeological site. I picked 8 of them. I tried to pick oysters that were attached to well, originally attached to archaeological material, because again, it was all repatriated, or looked like it had once been attached to archaeological material. So I tried to pick those. And I also had to try to pick ones that were pretty well preserved, because it's quite, you can't undo it once you embed it in epoxy and cut it in half. So I also sampled a few ancient barnacles. I wanted to see if the barnacles were forming at the same time as the oysters. Were they introduced at the same time? And then I also sampled 2 modern oysters that I collected from the back of Manasota Key, the barrier island, because I wanted to see how much the environment had changed over thousands of years. And you also kind of need something to compare ancient oysters to. So modern oysters are a good choice. And then finally, to check my homework, I collected a few freshwater snails from the peat from that pond. So theoretically, when I get my isotope analysis, my data back, the snails should look like they're in a freshwater environment and everything else should not. So that was more to just like have a baseline for the pond. Here we go. Okay. So this is what it looks like when I cross, or this is what the data looks like. I had 3 pieces of data associated with each oyster. And I mean, each oyster might have had like 30 pathways I milled. So tons and tons of raw data. I have the location along where that hinge is, like where I collected that sample. And then I have an oxygen reading and I have a carbon reading. So each oyster had 3 pieces of data associated with it. So again, I mentioned like over 500 pathways I milled in all the total organisms. So with all that data, the first thing I did was put it into a cross plot in Excel. So you can already see the 2 modern oysters are completely mapping completely differently to the archaic oysters. So the oxygen reading is on the x-axis, and then the carbon reading, the delta 13C, is on the y-axis. So that to me is indicating these two groups of oysters were operating in completely different environments. So, all right, proof of concept so far. Good. That's what we wanted. If we distill all of these data into means, so really just I took the mean oxygen value for an oyster that maybe I had milled 57 times, the mean carbon value that I had milled from the oyster. That's what's up there. So we can already see that the two modern oysters in terms of carbon are completely different to the archaic oysters. So something is going on here with carbon compared to the archaic and modern oysters. And then for the oxygen means on the right, we can see the modern oysters are the ones with the red box around them. And what was really interesting to me is archaic oyster values are kind of capturing a changing environment. So this was my first indication with just the mean of each oyster that they're maybe living in different environments over time. So super excited about that. Oh no. Can't go backwards. Okay. How does this compare to the barnacles and those freshwater snails? So I told you the freshwater snails better be living in a more freshwater environment than the oysters, and that's true. So my homework, I checked my homework and it was good. So on the right there, that's the oxygen environment, and we can see that the freshwater snails are way down here in the negative, so it's more freshwater. I was like, thank God, because if it was opposite, I did something wrong, and there goes a PhD. So, and again, with the mean value of carbon, you can see the modern oysters are way out here on their own. What does that even mean? This is unfortunately a result of burning of fossil fuels. So I can't really compare the modern oysters in terms of carbon to the archaic oysters anymore because we have modified our environment so much that, like in terms of carbon, we've modified our carbon environment so much, burning the fossil fuels, pollution, runoff. It's called the Seuss effect, that basically these modern oysters are going to just wildly be different in terms of carbon to anything pre-industrialization. So I can't really make different inferences anymore because of that. But I wanted to focus on oxygen anyway today. So, right, back to my cross plots, which I really like. So I cross plotted all the mean data. So mean oxygen and mean carbon for one oyster, cross plotted. You've got your oxygen value on the x axis still, and then your carbon value on the y. And you can see the different groupings. We have the modern oysters in the top left, capturing their modern oyster-y environment. We have the freshwater snails at the bottom there, capturing their freshwater snaily environment. And then we have the barnacles and the oysters, the ancient barnacles and oysters, capturing their environment. And as humans are wont to do, we like to group things together. I noticed some interesting groups of specific oysters compared to the rest of the data. And at first I was like, am I just trying to draw boxes around things? Am I trying to make this fit? But this gets really interesting when I compare it to radiocarbon dates. So I sent all of my samples off for radiocarbon dating. And what I was super excited about was there were two separate groups of radiocarbon dates. We had an earlier, older group, and then we had the later second group. Those top two dots, ignore those readings because that's from sediment. So I just didn't edit those out. But I was interested in these groupings because I was like, is this real? Am I making this up? Is this two separate instances? Have I answered my questions? But when I compare it to the isotope analysis, when I compare it to the results, the group 1 oysters are capturing a fresher environment than the group 2 oysters. And radiocarbon dating can be really complicated for a variety of reasons, especially for shell carbonate. So if you're up close, you might be able to see that the age of group 1 says 10,000 years old. They're not 10,000 years old. That's a false effect because we know the burials are like 7,000 years old. There's no way these oysters are 10,000 years old. So while I cannot trust the actual number, like age, I can trust the groupings because I checked my homework. I had an example of a small oyster that was attached to a human temporal bone, and inside of that oyster was a barnacle. So that means the oyster had to settle on a temporal bone first and form, and then it lost its upper shell. And then a barnacle at some point came later and attached to that shell. And I tested that, and it was right. So the oyster was older than the barnacle. Not 10,000 years, but so I can't trust actual age, but I can trust the groupings. So in terms of answering, like when were these oysters introduced to this pond, I still can't really say that. I can say it's after 7,000 years, but I can't give you an exact number, which sucks. All right, so to reiterate my biggest findings, based on all the data, the oysters actually revealed two separate time periods and two separate environments. These I am interpreting as inundation events. It may have been more subtle than that, I'm not sure, but I'm interpreting it as inundation events. The first group, which you can see there, they formed first in fresher water parameters. And it's also interesting to me that the condition of these oysters is a bit rougher. They're more deteriorated, they're kind of beat up compared to group 2 oysters. A little bit prettier. So group 2 formed second, later in time, in an environment that was more saline and at times hypersaline, which means saltier than ocean water, which is very interesting. Hypersalinity can occur in smaller bodies of water where evaporation is happening. So imagine water evaporates, leaves the salts behind, makes the water that's left even saltier. So the pond could have been somewhat isolated at this time, so it was still being affected by evaporation and, of course, precipitation and groundwater sources. But as the sea encroached and larger waves, we're introducing oysters, some evaporation happening, we get a hypersaline environment. I think the inundations, as I said, happened at least twice. There could have been more. I only studied 8 oysters. If I ran a couple more oysters through isotope analysis, maybe I'll see other types of environments and different dates. I'm not sure. But we do have estimated past shorelines that, because I told you I have no idea when these oysters were introduced. I can't trust the dates. We have some estimated past shorelines that can kind of be bookends. So there, I don't know if you can see those in the back, but the one farthest, closest to the archaeological site is 6,000 years ago. And then we've got 7, 8, and 9,000 years ago. So that's where the estimates of these shorelines would have been all those thousands of years ago. So based on this, we think the pond, this archaeological site, was fully inundated and like completely in an offshore environment by 6,000 years ago. It may have started around 6,500 years and just like took a while to be completely awash. But so the oysters couldn't have happened, before the burial 7,000 years ago. And it definitely would have been fully inundated by 6,000 years. So, right. So at some point, the oysters had settled on the human remains and the wooden stakes, and they grew for a time before the site was eventually fully inundated and maybe even capped with marine sand in like one event, perhaps during a storm. And I say this because the most recent oysters, those group 2 oysters, they all died during hurricane season in Florida. And I'm not getting into season of death estimates, but it basically is like looking at the color of the bands and comparing that to isotope data. But it was very interesting to me that they were dying during hurricane season, so I could totally make up a story in my head where there was just a huge storm event, brought in all this sand, capped the site, led to preservation, that kind of thing. So, but maybe that's reaching, I don't know. What is amazing is that delicate and very old archaeological material survived sea level rise. What I told you earlier was a very catastrophic process. Many archaeologists, my predecessors included, did not expect sites like this to exist in an offshore context, especially after human remains could have been exposed for years after oysters were attached to them. Remember, they need access to the water column. And when I did some age estimates of the oysters counting those growth bands, the oldest oyster I had was at least 3 years old. So at some point, human remains were exposed for maybe 3 years while this oyster was attached to it, which is just like remarkable to me. And I'm like, how did that happen? The secrets may be in hypersalinity. So with a lot of salt in the water, it kind of prohibits biotic growth. I mean, if you imagine making preserved foods, it's in a saltwater bath. You know, you put some like cabbage or onions in a jar with salt and water, and there you go, you got fermented food. So I don't know, fermented archaeological site. The other thing is continuously saturated environments preserve material a little bit better. So the worst thing that you can do for organic substances is like it's dry, wet, dry, wet, dry. That creates really bad deterioration. So I think the pond must have remained like fully inundated at some point enough to preserve the material. So yeah, hypersalinity and a fully saturated environment, and then maybe capping by a storm allowed preservation of such a sensitive site. Until now, unfortunately, due to like coastal development, it is eroding out of the seafloor. So that's why it came to our attention. So there were tons of other aspects of my research that I did not want to get into because it would be a 2-hour talk. And yeah, I'm not going to do that to y'all. But that's my general spiel about how I used oysters and isotope analysis to study a site that was once an inland mortuary pond and then slowly became swallowed by the sea. So thank you. Questions? Okay, yes.
Question. I missed the importance of the hinge.
Oh yeah, I was gonna wear my oyster earrings. I could have like brought that to you. Basically, the hinge is the biggest surface area. So if you imagine the oyster is kind of like growing, there's the top shell and the bottom shell, which is cupped, which has all the meat in it. So the bottom shell is usually bigger. And that's like where the base of the hinge is. And the top shell just kind of is like a thin cap that goes on top. So that's usually the one that falls off and gets lost in archaeological sites anyway. So what's left is this really robust hinge area. And thankfully for us, that's where you can collect those isotope, the powdered calcite, which is then what I use to make isotope inferences. So kind of like, I guess the whole tree, like the tree ring metaphor, the whole tree is creating rings. The oyster shell itself is really just creating bands in that little hinge area. So yeah. Yeah.
You mentioned towards the end there, the sites eroding because of coastal development.
Yeah.
You know, the awful things about coastal development. I'm not getting how that would cause erosion on coastal sites.
Yeah, so we think in a couple of studies after the Venice Inlet, or there's like a Venice Pier and the Venice Inlet was formed, that actually affected the longshore drift of sand. So instead of sand accumulating where it should, it changes basically like subsurface currents and where sand is basically being borrowed from and replenished. So and we've gone back and forth multiple times. We've gone after storms. And I always thought after a storm it would be like completely visible. But there was like one storm that we visited the site and it was like covered. And I was like, okay, that's good. But it is rapidly eroding. I mean, just because we started studying it in 2016, and then I was there in 2024, and we lost meters of peat, like just gone, and it's just breaking off and blown away. So yeah. I saw yours first and then I'll get to you up here at the front.
When you said the oysters were 3 years old, that means they lived for 3 years.
Yeah, yep. So
1,000.
yeah. So I don't know how old or like how ancient they are, but they were 3 years old when they died. So, but there were some that were 1 year old too. So I could see like material being exposed for a year. And age estimates can be a little complicated in oysters. Theoretically, in a perfect environment, they're laying down a gray band in the winter months and a white band when it's warm. So you get like a pair of bands is 1 year. But sometimes it doesn't always happen. And that can be a little tough when you're in subtropical areas where it's like always warm, and it can be related to when they're spawning because that's really stressful. So maybe they don't lay down a ring, so. Right.
Can you explain the pre and post industrial carbon or isotope? What are how do you identify what one versus the other?
Oh, okay, so it's just about the ratio of the carbon. So pre-industrialization, there wasn't as much carbon. Burning fossil fuels releases tons of carbon, and there wasn't as much carbon pre-industrialization. So now these oysters are taking up like lots of extra carbon. And that's also why radiocarbon dating is hard because it's taking up extra carbon and then it looks way older. And I'm like, that can't be right. It's not 10,000 years old. So it's not like necessarily determining like what's a pre and post. It's more of like, that's like that carbon value is so not like it's so far away from everything else that it's probably
So
pollution.
you're saying it's not linear, in other words, that there was a jump and so that.
Right,
they started.
I forget what is it like 1950 or something? There's like some
Yeah.
age or whatever where it's like, not 1950, I don't remember what it is, but there's like some year where it's like, well, you can't study carbon in like oysters after that because it's all messed with essentially. It's yeah, poison. We poisoned the well with too much carbon. But I could not get access to oysters that were pre-industrialization because there are some universities and collections that have pre-industrialization oysters and they use those as controls for when people are trying to radiocarbon date. And so if you have an oyster that was collected like in Tampa pre-industrialization and you're trying to date an oyster from your archaeological site, you can like correct, based on that master oyster, but they're not going to let me cut into theirs. So
Melissa, are the peat burials in Florida, are they roughly the same period?
yeah, mostly middle Archaic period. Yeah.
Middle Archaic.
So about 7,000 years ago.
So is there any evidence that this practice was transferred to other areas by communication between tribal groups or ethnic groups?
That's such a good question. So we know that water burials are significant in other cultures, I don't know what the significance of burying people in water here in Florida was, but I don't know if it transferred to other groups. Are you saying like within Florida, maybe the practice or
somewhere
okay, yeah, I'm not sure.
as far south as
Because you've got like cenotes that were really,
Yeah,
really significant.
Mexico.
Yeah, so we do have indications of water burials, and I also think like bog bodies in the UK, but I think that's more of like things are just falling in and becoming preserved. But what's unique about Florida is in the area where we find those mortuary ponds, like the Central Peninsula, the environment is so specific, and at the time, these ponds were just like peppered all over the state. I am kind of thinking these were mobile groups and one of the studies at Windover showed that people were buried within 24 hours. So I'm thinking like these ponds were all used in a very specific time period. Some of them were used for 500 years by these people, like over and over and over again. So maybe they are actually just moving around the state. They know where these ponds are. And if they lose someone they love, they know that, okay, there's a pond not too far away. We'll inter them there. Because I mean, yeah, if you're super far away from your Windover pond and someone's buried within 24 hours, so my theory is that they were actually had a network in their mind of which ponds do we use over generations and generations and generations as we're moving across this particular portion of the state. But I don't know, it's all conjecture. No idea.
Related to that, do you think that there was some knowledge of the preservation of the body that goes along with?
I think
bogs?
so.
Yeah.
Yeah, and I think that was like even suggested with the Windover burials. They were suggesting, the archaeologists who led the Windover excavations were like, okay, they knew the properties of the different types of wood that they used as wooden stakes. And they probably knew the preservation qualities of like these waterlogged boggy types of sediments. So yeah, I think that's absolutely like possible. And there were some objects in Windover like shell pendants and tools, like bone tools and faunal bone. Unfortunately, because we had the bases of burials, we only found like 3 examples of worked shell and 8 examples of tiny pieces of cordage, which we were shocked was even preserved. It was like that big, and it was like spun cordage from saw palmetto, I think, is what they had woven together. So.
How did you date? You said it's 7,000 years old, but how did you come to that date line?
Yeah, so we We dated wooden stakes. So we don't date human remains out of respects for the human remains, but also out of respect for the tribes. So I mentioned all of the material had been repatriated. But originally, when we were trying to determine if this was a mortuary pond, we dated a tiny piece of a wooden stake, and then I think we dated two other tiny little fragments of wooden stakes just to get those solid dates and like, okay, now we know what we have. We don't have to like date anything else. And that's also kind of why I wanted to study the oysters, because it's an example of how you can do archaeology, often a destructive process. Like there is a way to study sea level rise without destroying the other archaeological material. So these oysters weren't archaeological, they were environmental, you know, so I don't have to disturb like a skeletal material or modified wooden stakes. Like I don't have to break those apart to study sea level rise. I can do something else, you know, I don't have to destroy sensitive material. So that was also kind of like why I chose the avenue of studying oysters instead. So
How did you date the states? With radiocarbon dating?
yeah, radiocarbon dating, we use a lab up in Georgia. So we've sent all of our material up to them, even for other archaeological sites, because they've got like really good standards and we trust their dates and we wanted to be absolutely certain. And I've dated like faunal bone, just like turtle bone and stuff from the original pond, which I had also included in my dissertation, just because I was like curious, how old is the like fauna that are coming out of the pond compared to the oysters, compared to, you know, sediment cores. So it was a lot. All right. I think that, oh, we had some layers. I saw your hand first and then we'll come to you next.
Is your research being applied also?
Isotope analysis is, well, mine specifically, I don't know, maybe, but isotope analysis all over the place. And when I was trying to do this study, we weren't actually sure it was going to work. So isotope interpretation can be difficult in oysters, just like radiocarbon dating. I picked the hardest thing. So this was kind of a test to see if we could get valuable data on ancient oysters. The guy that I worked with in Alabama, he was on my committee and he was like, wow, we're mostly studying like more modern materials. So he was like nervous for me because he was like, what if nothing happens and all the like data is wrong? But it worked really well. So we actually published some articles about it because we were like, hey, you can get valuable information from ancient oysters if you find them on your archaeological site, you know. So that, and then also we weren't sure how well the freshwater snails were going to I wasn't sure the quality of that data either because they're very old and these gastropods are like, you know, amphibious. So there were some equations that I left out, but there's like basically a paleo temperature equation that I had to use to like get an idea of how saline water was. So yeah, a lot of inferences and I wasn't sure it was going to work at all, but it worked enough. And then your question.
Yeah, following up on that, what is the current research that's being done in our area now, underwater, basically archaeology underwater?
Yeah.
Because I think that has basically expanded
Yes. Yeah.
based on what I've heard.
It has definitely, and it's sites like this when people said there's no way that exists, there's no reason to look for those. Like if we're gonna go put an oil line in offshore, we're just gonna look for shipwrecks. We don't need to look for anything else. This site proves you actually need to look for other things because there may be preserved really old sites that will tell us when humans were in America that are preserved in these continental shelf areas, like the offshore continental shelf. So there's been an explosion in what we call submerged pre-contact archaeology. So pre-contact, like pre-1492, before contact with Europeans. So there's been an explosion in studies of that, and Florida is a great place to do underwater archaeology, especially for submerged pre-contact sites, because we have great preservation in rivers and sinkholes and offshore. So in Appalachian Bay itself, that has the highest concentration of submerged pre-contact sites based on surveys that folks had done in the early 2000s. And they found that if you look for submerged pre-contact sites, you often find them. So yeah, anytime someone tries to tell me, yeah, no, that stuff isn't preserved offshore, I'm like, that's cool.
Who is funding that research
Mine or any in general?
in general?
It depends. The Department of State actually gives grants to fund research for offshore survey. BOEM, Bureau of Ocean Energy Management, is deeply invested in this because they're the ones who manage offshore oil and gas and even wind farms. So you imagine if they're trying to conduct a survey before they build these giant structures on the seafloor, they have to basically say, we're not going to put it through a shipwreck. We're not going to put it through a submerged pre-contact site. So a lot of those studies are funded by BOEM. There's like 2 going on right now because they're recognizing that there's standard survey. So if a guy wants to put a wind farm in or something, he has to do a survey first, an archaeological survey, and has to follow their standards. And they're realizing their standards aren't actually catching submerged pre-contact sites. So they're doing studies now. And they funded some of our research because they realized, like, we're going to do a test survey to the BOEM standards over Minnesota Key offshore and see if it catches it. And it didn't. So, yeah.
So, one of the things I like about SciSalon is that it's about communication, and not just the findings, but the ability to talk to people about it. So what I wish I could ask everybody who does this, I know it's probably like a lot of different things, but can you name one thing that happened that in your education that made you want to go in this direction? Like a person that you studied with or
Okay. Yes, actually. I was a terrestrial archaeologist originally. I've always just been fascinated. I thought I was going to go into anthropology, and then I was like, I don't want to talk to people. I'm going to dig through their trash. So I got into archaeology, and I was taking like some archaeology class, and I think the professor was like tired, and he was like, we're going to watch this Titanic documentary. And it was basically these guys were just like collecting samples, and he was like, yeah, underwater archaeology is pretty cool, right? And I was like, that's a thing. So that's honestly just in passing. He mentioned underwater archaeology, and I was like, I want to go deep, dark, and scary. So I marched up to my advisor's office, and I was like, I want to study underwater archaeology, and she laughed at me. So then I was like, okay. So I switched advisors, and then I headed out. Yeah. It's amazing what one person can like influence you. You know, you never know. You might say something and it changes the person's trajectory of their life. So, yeah.
Just an add on to what Eric just said about science communication. My background is in environmental communication, a lot of science communication in that field. And I just want to say what a great job you did of explaining the very technical topic.
Yeah.
Like be those tools.
I'm also clueless because isotopes were not my background, so I had to like panic learn all this stuff as I'm in the lab in Alabama, you know? So thank you. Thank you guys.